To welcome everyone. Welcome to our online visitors to our second in-person and online hybrid meeting of the Linnaean Society. Remember that email that went out reminding you to pay your membership dues. If you haven't done it, click on the link. We use those funds to support events like this and other events that we have coming up. Our fall field trips are still going strong. In fact, you just missed the registration deadline for Birds and Brews about an hour ago. That's going to be in Greenwood, but there are other trips. Look them up on the website. Next month's meeting happens here in person at the Liederkranz Club, and it's going to stream online as well. It's back to Tuesday, November 11th. The society, we still have an open position for president. And so if you know someone who's interested, email the secretary, get in touch. Until then, the board is rotating as presidents. I'm up this month. Next month, it'll be Peter Davenport, who probably greeted you and got you a name tag in the back. And we thank him for that. So we have also almost a dozen new members who joined in the last month. If you're in the room, you can feel free to wave, stand up, introduce yourself. So the new members are Robin Daly, Beverly DeVoe, Trustin DeVoe, Elliot Jenkins, Bruce Liebstone, Patrick Marcoux, Jane Markey, Catherine Pangiordano, Kimberly Schwab, Wendy Spence Schmidt, and Terry Von Plennies. So welcome to all our new members. Thanks for joining. And sorry if I mispronounced any names. Our Vice President, Karen Becker, has put together a great program tonight. Our presentation will be from Dr. Richard Lankau, who is a professor of plant pathology at the University of Wisconsin-Madison. His talk is titled, Forest Fungi and Forest Health. What do we know? What don't we know? And how can we use this knowledge to preserve and restore healthy forests in a changing world? Please, let's welcome Dr. Lankau.
Thank you. I want to thank Karen and the board and whoever else was involved in the decision to invite me out here. It's only my second trip to New York City in my life, so it's been fun. So when Karen emailed me, she said that there was some interest in learning more about what's happening in these forests that you're going on all these trips to that you might not be seeing, especially if you're looking up to find the things that fly. So what I want to do today, as you are all aware, forest, they cover our world, something like.
Can you stand in front of?
They can't see my face. Okay.
Speak directly into that.
Okay, is that better for try to stop my wandering. It's a habit. So forests cover a large chunk of the world, 30% or so of the land surface area. And just in addition to being beautiful, they're incredibly important, right? They drive a lot of the functioning and the cycling of our whole global system. And so when most of us go out into the woods, we see this, right? What I want to sort of build an appreciation for tonight is to start thinking about all the stuff that's happening below your feet in that forest. And I want to point out that the woman there on your left, Dr. Cassandra Alsop, is my partner in crime for all of our work with forest and fungi. So I may forget to say every time our joint work comes up, but pretty much everything I talk about today, we've done together. And so there's a lot going on below ground. And this, we just meant to show like there's a lot of biomass below ground, there's a lot of those roots of the trees. And there's a whole lot of stuff going on that you might not be able to see. So all of the pictures up here are examples of different kinds of fungi that are doing different things to affect how a forest functions. And I know this might be old news to some of you, but wasn't sure what everyone is bringing to the room tonight in terms of how long it's been since you were in a college or high school biology class. So let's start a little bit with, what are fungi even? And if you think historically how scientists have even thought about fungi, it's changed a lot. They used to be considered kind of weird plants and were studied by botanists. But as we learn more about the real evolutionary history of the world, we get better tools to understand who evolved from who and how things are related. We get a very different picture of Earth's history than you might just by looking at how things look to your eye. And so this is a phylogenetic tree. It's meant to map, all of the biodiversity on the planet and who's related to what. You can see my cursor there. All of this up here, which represents the vast majority of evolutionary history and genetic diversity in the planet, this is all bacteria. This is all single-celled organisms. They don't have a nucleus. They had this planet to themselves for like 1 billion years before anything multicellular came about. And then of what remains, all of this, which is the most of what remains, is this group we call the archaea, which is a group we didn't even know existed until 30 years ago. They look pretty much like bacteria. At first, we only thought they lived in like hot springs in Yellowstone National Park or like deep in the ocean vents. Turns out they're also everywhere. They just don't grow on petri dishes, so we never knew about them. And they're invisible. So together, all of this and all of this is just these unicellular, tiny little cells, bacteria and archaea. They're really driving the world. And they're the vast majority of genetic diversity in the world. Everything you can see with a naked eye is in this little green slice here. And it's not even most of that green slice. So those are our eukaryotes, so the organisms that have nuclei in their cells. And now if I blow that out, here's the diversity of those eukaryotic organisms. Plants, recognize those? Those are just this little tip here. Animals, all of animals, this little tip here. And fungi. And what I put all this stuff up here to point out is that fungi, if you look at the way you read these trees, it's like a family tree, right? You sort of walk back through the line to see when something most recently had a common ancestor. So fungi, despite what the early botanists thought, are not weird plants. They're actually much more closely related to animals than they are plants. And that's really important to understanding what they do and why they do it and why they might be important. Because the really key similarity between animals and fungi is that they both have to eat. They have to consume organic material to get their food and their energy. Plants, what can plants do that no one else can do? They can make their own food from sunlight and air. So this drives, the evolution of fungi, it drives their lifestyles. What is different about fungi than animals, I often describe to my students, fungi are kind of inside-out animals. So when we need energy and food, we eat it, it goes into our stomach, and we digest it from the inside, right? Fungi don't have stomachs. They exist as these unicellular strings in the world. So they actually do the opposite. Their stomach's on the outside. They exude enzymes out into the world to digest their food outside of them and then suck it up as the little broken down pieces. So it's back to forest, thinking about, what are fungi doing in forest? There's 3 major sort of lifestyle roles I'm gonna focus on. And they all relate to, again, how is that fungus getting its food? How is it getting its nutrition and its energy? So one really key role for fungi is to be decomposers. So decomposers are any organism that's getting their food from dead organisms. That could be very recently dead organism, something that just died yesterday, or it could be the remains of a dead organism that are hundreds of years old, right? So living off that organic material that's breaking down in soils, the humus in soil, your organisms are living off that. So those are your decomposers, very important. I'll talk a little bit about them. As you're probably aware, there are a lot of fungi that get their energy and their food from living organisms. And if they do that to the detriment of their host, we call them a pathogen. So a pathogen is something that's getting its food or its energy from its host, and that's hurting the host. That's doing nothing good for the host. And you can contrast that with this broader group that I'll call mutualist. The word, you might guess, comes from mutual, so mutual benefit. So these fungi are also living off a host. They're getting their energy and their food from their host, whether that's a plant or an animal. But in the end, the host is doing better because they're there. There's some kind of a relationship where whatever the fungus is taking from that host, it's more than paying it back in some other service that's making the host healthier and grow better. And so that's what I'm going to spend a lot of time talking about tonight, are these mutualistic relationships between plants and fungi. And it's not just plants. There are fungi that live on animals. There are fungi that are pathogens and parasites of animals. One thing I won't go into much, fungi are the major pathogens of plants. So if you're a gardener, you probably fight some diseases in your garden. They're probably fungal diseases. If you're a farmer, I work with a lot of farmers in the Midwest, we're fighting fungal diseases all the time. There are some bacterial diseases of plants, there are some viral diseases of plants, but fungi are the primary ones. Which is very different from humans, right? There are fungal diseases of humans. They're relatively rare in the grand scheme of things. They often can only really cause problems in people that are very immunocompromised for some other reason. And that partly has to do with the fact with how similar fungi and animals are to each other genetically, how different fungi and plants are from each other. Fungi have an easier time invading plants for a variety of reasons. So just to go into a little bit of that background of those different roles, that role as decomposers. These fungi are forest recyclers. So fungi, along with all those bacteria that I mentioned, that was the vast majority of life on Earth, they're vital for breaking down dead material, especially dead plant material, in order to recycle the nutrients. So if you think about what goes into making your body or a tree's body, we're all made of the same stuff, right? We're made of carbon, primarily, hydrogen and oxygen from water. And then we need nitrogen. That's a component of protein. We need phosphorus. So these are the elements that limit the growth of life on Earth. Plants need all those same things, but plants can get their carbon from the atmosphere, right? They suck up carbon dioxide out of the atmosphere. They combine it, use the power of sunlight to combine that in the water to make sugars. We all depend on that. But they still need to get those other elements. They need to get nitrogen and they need to get phosphorus. And plants are actually worse at getting those things than we are because they can't digest. They don't have stomachs. They don't make the enzymes that fungi make in order to digest things. So they have to suck up nitrogen in really chemically simple forms. So again, if you're a gardener, if you are putting plant food on a garden or a house plant, what's in that plant food? It's three things primarily: nitrogen, phosphorus, and potassium. And those are the three things plants need from soil, in addition to water. In order to access that nitrogen, phosphorus, and potassium in nature where no one's fertilizing them, they need to recycle it. It needs to come out of that dead material that's in the forest floor. And so that's what fungi and bacteria are incredibly important for. Without them, a plant would grow, a tree would die, and then it would just sit there forever, right, as a log. And all of the nitrogen and phosphorus and potassium that went into building that tree would be forever locked up in that tree, never to be used again by another plant. So we need these decomposers to break that down. And the fungus is just trying to make a living, right? They're trying to get after the carbohydrates in that dead material, and they're getting the energy from there. But in the process, they're releasing the nitrogen, the phosphorus, and potassium that's allowing the other plants in the forest to grow. Fungi in particular are somewhat unique in the ability to break down wood. So wood is, as you might imagine, very tough, hard to eat. Not many things make a living by eating wood because it's chemically really complex. And so there are only a few kinds of millions, but compared to most life on Earth, it's a rare trait for an organism to be able to break down wood. And it's a trait mostly limited to fungi, the white rot and brown rot fungi, which we know a lot about because as vital as they are for a forest, they're also annoying when they're decaying your house, right? These are the same fungi that are breaking down the lumber in buildings. But white and brown rot fungi are great for the world because they're out there in the forest decomposing wood. I wanted to highlight throughout where there are places where the scientific community is really actively trying to learn new things. This role of decomposers in forest is actually a really active area of research because one of the biggest questions those of us who think about climate change have to grapple with and are able to sort of predict how is climate going to change into the future. We know what's happening with carbon globally in the world, all this carbon in the atmosphere that we're pumping into the atmosphere, right? Is it going to stay in the atmosphere? Is it going to get pulled down out of the atmosphere? Plants pull carbon down out of the atmosphere, right? That's how they grow. And then that carbon is in a plant for some amount of time before that plant dies. And then what happens to it? Is it going to get recycled back up into the atmosphere? Or is it going to get stored in some stable form? And where that carbon is mostly stored around the world is in soil. Soils are our biggest pool of carbon. Most forest around the world, we think, most of the time, forests are what we call a carbon sink, meaning that there's more carbon going out of the atmosphere into the forest than there is carbon coming out of the forest into the atmosphere. It's a dynamic mix. Everything's breathing, right? Plants breathe too. They also breathe out CO2. Animals breathe out CO2. Fungi and bacteria, when they're doing their stuff, they're breathing out CO2, just like we are. So there's carbon dioxide coming out of the forest. There's carbon dioxide going into the forest as the plants grow. The balance of that determines whether this is a sink. It's making the problem better for us by absorbing all that excess carbon dioxide that we're putting up there, or is it making the problem worse? So there's actually more carbon coming out of the forest and going in. As I said, most forests, most of the time, we think are sinks, but not always. So as conditions change, as things get hotter, as forests burn, the giant wildfires that you have out west, as forests are logged, all the things can change a forest from being a sink to a source. So we're keeping a close eye on trying to understand what controls that transition. And these decomposer fungi are a big part of that. Are they becoming more active? They're decomposing faster. It's going to make it a source. Are they slowing down? Are they storing the carbon in some other way? So I mentioned that fungi can be pathogens, right? So it's another really important role they have in the world. And it can be devastating. Many of you may be aware, this part of the country, right here, 150 years ago, you would have been in the center of the range of the American chestnut. Right? That would have been one of the dominant trees in the forest around here, would have been the dominant tree all over this part, North America. And then in 1910, an exotic fungal pathogen was introduced. The causal agent of chestnut blight. And this is just a map of the spread of that disease in 1910. And 30 years later, it spread across the entire range of the chestnut. And now you would be very hard pressed to find a chestnut tree. And if you're here in New York, if you do find a chestnut tree, it's going to be small. But if it gets about this big around in its trunk, it'll die of the blight. The tree is, we say, functionally extinct. It's not completely extinct. We have essentially museum specimens of the tree growing. There's a patch where I live in Wisconsin where they haven't gotten infected with the fungus yet. But as a component of an ecosystem, it's gone, right? And that's a consequence of this fungal disease. But since I'm being a cheerleader for fungi tonight, I don't want to dwell on that. Even fungal pathogens get a bad rap. Most pathogens, most of the time, are not driving their host extinct. That's a bad strategy if you're a pathogen, right? Pathogens are actually vital to maintaining the diversity of a forest. So how many different kinds of trees growing there, how many different kinds of trees you have growing in your forest, then cascades down to affect all the different kinds of whatever else is living in that forest, right? Because most plant pathogens are specialists. They only infect one or a few species of tree. And this is a little seedling of a cherry tree dying, of what we call a damping off disease. So that's a fungus that's infected its roots and killed it. But this fungus only infects cherry trees. If there were an oak tree living next to it, it would be fine, or a maple tree, or anything else. And so what happens is these fungal pathogens create zones of death around sort of the maternal tree. So you have a big adult cherry tree, or in a zone of soil around that adult cherry tree, none of its seedlings are going to do well because it's going to be loaded up with these fungal pathogens of cherries. The oak tree, same deal, but it's a different fungus. So it's killing oak seedlings. The cherry seedlings can live underneath the oak tree. The oak seedlings can live underneath the cherry tree. You put that all together, and you have a system where no one kind of tree can take over. And so we think that's the primary reason, or one of the primary reasons, why you can get lots of different trees growing together in a forest. Why in the tropics you can have 300 species of trees in just a couple of hectares of forest. There's no good way to explain that other than through the action of these specialist pathogens. So even pathogens have some role in the world. But I'll talk most about these happier mutualistic relationships between fungi and plants, where the fungus is benefiting because it's getting its food from the plant, but the plant's also benefiting in some way. And how the plant is benefiting actually can be pretty diverse. There are a bunch of different kinds of fungi that can do something good for plants. Some of them live in the leaves of the plant, some in the stems, some in the roots. They can be helping the plant acquire those soil nutrients. They can help plants tolerate stress like drought, heat, or cold stress. And again, this is an area where this is a really active research area. How is it? Why are these fungi? How are they doing it? How are they making a tree better able to survive cold or better able to survive heat? It's a little bit mysterious still. And then the other thing, lots of these endophytic fungi, so the word just means lives inside plants, they can defend it against those pathogens. So plants often rely on other microbes to defend them against the pathogenic microbes because their immune system does not work the same way ours does. So these mycorrhizal fungi, these beneficial fungi, well, this is one category of beneficial fungi that some people may have heard of, maybe a new word. It's Greek. It just means fungus root, where the word comes from. So it refers to this joint organ where the fungal tissue has grown inside of the plant tissue, sort of living together. But the fungus also grows out away from the root into the soil. So they've got this dual life cycle where they're living inside of plant roots, but they're also living out in the soil, and they're a connection from the root to the soil. Two major groups of these fungi that do this. These are arbuscular mycorrhizal fungi. This is a really ancient group. I'll show you some fossils on the next slide. That is in almost all plants. Then this ectomycorrhizal fungal group is newer. It's like the new innovation. And by new, I mean 50 million years old instead of 400 million years old. And I'll talk a lot about this because this, although it's newer and kind of rarer, it's really important in forests specifically. So they're morphologically different, but they're also functionally different in terms of what the fungi can do for plants. These are often going after primarily phosphorus, and they're kind of like straws. They're little tiny straws that go out into the soil and suck things up and transfer to the plant. These fungi grow into the soil, and they can be a little more active than just being straws. They are, where I mentioned how fungi can like digest from outside in. they can exude these enzymes into the soil to break down organic material in order to liberate the nitrogen that their host plant needs. They're out there, what we call mining for nitrogen out of organic matter. Again, that's something plants can't do on their own. So if you're wondering, if you're wandering around forest, most plants you see, any kind of herbaceous plant, any grass, any kind of crop plant, almost always they're going to be forming these arbuscular mycorrhizal connections. There are only 3, I think, something, we don't know the exact number, we haven't looked at every plant in the world, but somewhere between 2 and 5% of plants don't form mycorrhizal connections. So it's by far the minority approach as a plant to live without fungi. The vast majority of plants are living with fungi all the time. Most of them with this arbuscular mycorrhizal group. And so tree-wise, maples, elms, cherries, ashes, cedars are some of the major trees in the forest around here that would be doing this kind of relationship. The only thing I want you to get from this slide is that plants and these arbuscular mycorrhizal fungi go way back. They evolved about the same time. So we actually think that the very first plants to climb out onto land from the ocean, again, over 400+ million years ago, did that with the help of these fungi. Although the word means fungus root, they actually predate roots. So the very first plants to grow on land didn't have roots. They evolved from algae. So they were like mosses, essentially. And so you think of what a root does for a plant. Plants need those roots to get water, to get all the good stuff in water, like the nitrogen and the phosphorus. When you're an algae in the ocean, not a big concern, right? You're floating in it. But when you're the first moss to live on rock, and you don't have any roots, it's a tough thing to do. And we think the only way they were able to do it is by bringing these fungi with them. I like to show this picture in my classes. So on these pictures over here, were taken in modern day, last year sometime. This is what the fungal structure looks like inside of a plant cell. So here's a fungal hyphae, which is their stringy cells, and they go into the plant cell, and then they start dividing smaller and smaller, and they make this, it's supposed to kind of look like a tree. That's how they get their name, arbor, like Arbor Day, arbuscular. They make these structures that look like trees inside the cell. So this is modern day. This is what's happening right now. These images are fossils from the Rhyolite chert. This is a mud flat that thinks really fine structures got fossilized, and you can even see cellular structures. And so if you look here, here's the fungal hyphae growing into a plant cell, making those connections. So this relationship has been around for a long time. That's my only point. And essentially unchanged. It looks very similar 400 million years ago to what it does today. It's in the other group of fungi. Again, I said it's rarer, but it's not that many species. It's only about 3% of plant species. But they're kind of important ones when you think, especially if you care about forests, because they're oak trees, hickories, cottonwoods and aspens, and then all the conifers. So pine, spruce, fir, all of those trees, which cover a large part of the world, are forming this ectomycorrhizal relationship. And so this is a different group of fungi, and it's a lot of different kinds of fungi. It's newer, as I said, only 50 million years old. And so the consequence of that, it's not quite as intimate. So the fungi are growing sort of in and around the plant cells, but not inside of them. And then they form these sheaths around the end of the root, called a mantle. I flew in earlier today, and I went to the American Museum this afternoon while I was waiting for my hotel to check me in. And they have a nice display in there. Next time you're in the History Museum, packed away in like one of the sections on like the natural history of New York or something, away from all the dinosaur bones. They have a nice diorama of like a below ground image of a forest, and it's got some great examples of these ectomycorrhizal relationships on oak roots. So it's kind of one thing I find really interesting about this plant fungal relationship is that it shows really clear patterns across the world. So this is a map of the biomes of the world. I don't know if you guys can read the fine text there, but this is really dividing all the ecosystems of the world into types. If you look at what plants are dominating these types, all of these areas, so your tropical rainforest, your savannas, your grasslands, deserts, all of those are dominated by plants that make this arbuscular mycorrhizal connection. So notice that's all around the equator and all the warm zones. If you go up high in latitude, what we call the boreal forest or the taiga, those are those conifer forests that cover Canada and they cover Russia and a lot of Northern Europe. These are all, almost 100% of the trees are forming these ectomycorrhizal connections. Where we are, which is right here, right, this kind of light green zone, called the temperate forest, also in Europe, also in China, in Asia, and a little bit in Australia. We have this mixed strategy. So you can find both. I mentioned some of the trees, right? Maple trees are doing the arbuscular relationship. Oak trees are doing the ectomycorrhizal relationship. And they're growing right next to each other in many forms, right? So that's kind of unusual on the world. Usually one or the other strategy is best, essentially. And it comes down to, I mean, if you haven't noticed the pattern, if you're in a hot place, you want to use the arbuscular mycorrhizal fungus. If you're in a cold place, you want to use the ectomycorrhizal fungus. Why is that? And why do we care about this? Why has there been so much interest scientifically in this? Is because it's not just a relationship between one tree and one fungus. It's actually a whole system. And the trees and the fungi, sort of their combined effects shape the entire way that forest functions. The way that energy is transferred through the forest and the way that element, or the matter, cycles through the forest. So when you have these AMs, arbuscular mycorrhizal relationships, so those kind of trees, your maples and cherries and such, in your tropical rainforest, it's an integrated system where the trees make their leaf, their litter is easy to decompose, juicy as we call it. So it falls, it decomposes quickly. The free-living decomposers, the other fungi, the bacteria can chew it up, release all that nitrogen. So we have what we call an open nitrogen cycle. Everything's moving fast. Nitrogen's flowing, it's cycling really quickly. The trees get what they need. It's not hard for them to get nitrogen. If anything, they end up getting limited by phosphorus because phosphorus doesn't flow in soil easily. You get a lot of bacteria, more bacteria than you do fungi. Potentially, and this is again, active research zone area, potentially as a consequence of that, you're storing less carbon in the soil. So again, why do we care about this? Because we're trying to model what's happening to the carbon cycle on Earth. As we add more carbon into the atmosphere, where is it going to go? Is it going to stay in the atmosphere and cause problems? Or is it going to get sucked back down into the biosphere? These kind of systems, we think, are doing a little less of that sucking carbon out because everything cycles so quickly. The carbon gets respired back up. Then over here, now imagine that you're up in like Canada, right? In a spruce forest or a conifer forest. Everything's slower, mostly because it's colder, right? So nothing's happening for large chunks of the year because everything's frozen. So if you've ever walked in a northern forest, feel like you're walking on the moon or something because there's this big layer of needles, the duff, they call it. So that's stuff that has not decomposed yet, right? So it's falling and it's just accumulating over the years because it falls faster than it decomposes every year. So it builds up. All of that represents carbon that has not been cycled back up into the atmosphere. Right? Whereas, I don't know if any of you have had the chance to travel to the tropics. If you walk around in a tropical forest, you're going to be walking on dirt, right? There's not a layer of leaves or needles there in tropical forest because all the leaves, they make just as much leaves every year. The productivity is high. But the leaves that fall get decomposed very quickly. And the rate of decomposition is faster than the rate of production. So you don't accumulate things. And so because it's cold, because the needles that pine and conifers make are hard to break down and they accumulate. That means they talked about all of the good stuff in those needles, the nitrogen that the trees need is locked up because it's not decomposing. And so that's the situation where those trees, in order to survive, evolved to partner with fungi that can grow into that stuff and mine the nitrogen out of it for them. So that's what these ectomycorrhizal fungi do. They grow into that layer of duff, and they can, with their enzymes, sort of specifically liberate the nitrogen, suck it up, and feed it to their host. And so the plant doesn't have to wait for the natural decomposition process to happen. And they leave the carbon behind, which a decomposer wouldn't do. Decomposer needs to eat that carbon and respire it. So these ectomycorrhizal forests, we think, are where we're going to get the big carbon sinks, because the plants grow, they suck in carbon, they drop it as their leaves, and then it stays there because the ectomycorrhizal fungi go in and get the nitrogen out of that for their host and leave the carbon behind. So it stays there and accumulates year after year. That's the theory. It's a little bit, there's some speculation in what I just said. There's some well-documented fact and there's some speculation. And so, again, why do we care about that? Because nothing ever stays the same, right? So where those spruce forests are growing, where those tropical forests are growing is changing as climate change. So this is just a map, of our neck of the woods, as we say. The red zones are where over the last, I think this was over 40 years, where we've seen an increase in the number of trees that make the arbuscular mycorrhizal relationship. Blue areas, like up here, where we've seen an increase in the number of trees making the ecto-mycorrhizal relationship. A lot goes into this. Since it all climate change, I think, has to do with warmer winters and the northward spread of some of these sort of temperate species, the maples and stuff pushing in our conifers in Wisconsin, where I live, being pushed up into Canada. But a lot of this is fire, deforestation, reforestation, abandonment of agriculture, suppression of fire, a lot of things going on. But there are a lot of people, myself included, that are really interested in this question and trying to figure out, what does this change mean for that question about what's happening to the carbon we put into the atmosphere? Is that making it suck up more carbon, or is it making the forest suck up less carbon? And what are those feedbacks? So the scary thing as a scientist who studies climate change is understanding the potential for positive feedbacks, where as we put carbon in the atmosphere, it warms up the world. That actually causes the planet to release more carbon. Right? So the biggest example of that is in the permafrost in the Arctic. We warm enough, there's tons and tons of carbon stored in the permafrost. If that stuff melts, all that carbon goes up in the atmosphere. And it's, really changes the game in terms of how much carbon we have to deal with. The other thing I thought I'd talk about, some of you may have been, if you have heard anything about mycorrhizal fungi, which I know most people haven't, but if you have, you might have heard about this phenomenon of the common mycorrhizal network. This is also sometimes referred to as the Wood Wide Web. And it refers to this phenomenon where that fungal organism that's connected to a tree, that same fungal organism could be connected to another tree, right? And that makes that bridge, it makes a connection between trees. And so there's been some interest in what are those, beyond just everything I've told you about what these fungi can do, is the fact that they're connecting multiple trees together in some kind of network have any extra special consequences. So there's been proposed that in those connections, there could be transfer of things between trees, transfer of water, transfer of nutrients, transfer of carbon as sugars, so that's essentially feeding other trees. There's some work on chemical signaling where, like, if one tree gets eaten by a bug, some signal transfers through the network, and then another tree is like more defended against bugs. It's fun to think about. It makes for great, like, puns. Where it's been most well studied is out on the West Coast in the conifer forest out there, so these Douglas fir forests. This is a picture of what one of those common mycorrhizal networks you might try to imagine what them looking like. So all of these green things are the trees. Then the little purple and blue blobs is where they think the fungal individual is. And they're trying to connect, the lines there connect sort of which trees are connected to which fungal blob. This is all done with the, essentially by DNA fingerprinting, like what you do to figure out who the murderer is at a crime scene. They're digging up roots and they're digging up fungi and they're trying to figure out are they genetically identical, this sample to this sample. And that's how we show that they're the same genetic individual. I do want to point out the caveat here is that the physical network is actually just inferred. It's actually almost impossible to know, is there actually a physical string of hyphae connecting those two trees, or is it just the same genetic fungus on each tree that has long since broken up in between them? That would be, as I say, almost impossible to prove. These strings are one cell wide. You can't trace them. But so I did a little bit of digging in, like what do we really know about this wood wide web thing? This question of can fungi connect multiple trees? Absolutely. That's been well documented in laboratory studies. We can make it happen between plants in a pot. Does it happen in nature? Well, very probably. But I wouldn't be a good skeptical scientist if I didn't point out that it's actually almost impossible to document it, that it's really happening, or to what extent it's really happening. But there's no reason to think it's not happening. Happens in the lab, probably happens in nature. Same idea with that. Can nutrients and water move across this network? That's been documented well in laboratory studies where we can really lock things down and make sure it's happening, use our isotopic tracers to say this nitrogen atom went from this plant through the fungus to this other plant. Again, does it happen in nature? There's some good suggestive evidence that it can be, but there are also alternative explanations that could have explained that pattern, right? It's hard to do the definitive experiment. What got to be more controversial with this among us sciencey types, was this question about moving of carbon. So because that gets at whether one tree is actually feeding another tree. It's one thing to share nitrogen. All trees need to get nitrogen. But one of the claims that came out of this work early was that adult trees were actually feeding sugars to seedlings and like helping them grow and survive those early stages through this mycorrhizal network. That's probably been the most controversial claim. That's not something we think of plants doing, feeding other plants. So looking up sort of what the evidence is out there for that, it's possible. Again, there's been some lab studies that are suggestive of that in the right conditions. Is it important in nature? Hard to say. Seems like if it is happening, it's at a really low level. And then some of the sort of more popular science aspects of this kind of went far ahead. The cart went far ahead of the horse. What do you say? I think. I don't know if anyone's heard of the concept of the mother tree. So this is the idea that trees were actually specifically feeding their own offspring to support them through their joint network. And that has not been really demonstrated. And I put unlikely there just because it's not clear how that would work. There's no good sort of plausible explanation for how trees recognize each other. Plants don't really recognize plants. You can take two genetically different plants and smash them together and they'll literally grow into each other. That's how we graft roots and around things, right? So plants are pretty bad at recognizing themselves. They're willing to mix roots up with a completely different unrelated plant, sometimes even an unrelated plant species. So it's a little hard to see how they would be finding their own offspring out in the forest of seedlings. We don't have a mechanism for that. And there's not a lot of evidence that's happening. So sometimes we have evidence of things that are happening and we don't understand how. And that's how science works, right? And then later on, we figure out how it's happening. This one I'm a little skeptical of, but that's sort of where that sits now. But I don't want to be dismissive. Even if this stuff ends up happening, I could be wrong. I could definitely be wrong. But even if I'm right and this stuff is really happening, this stuff could actually be really important. That movement of water and nutrients, especially Again, from adult trees to seedlings in drought conditions, that might be a reason why seedlings are able to hang on. Because being a seedling is the toughest time in the life of a tree, right? Once you're a big giant tree, you're pretty stable. You can handle a lot of things. When you're a baby tree, you're pretty vulnerable. So anything that helps the baby trees get out of that stage could be important. Okay. I'm glad we have long enough up because I have rambled on more than I thought I would. I want to spend the last bit, I won't take too long on it, is talk a little bit about what my lab specifically is doing, and try to get more of the why is all this stuff important to know about? So yeah, forest, I'm sure you're all aware, they're under threat, right? They're really important for the world. They're sucking up carbon. They're doing all the things, but things are happening to them. And so we need to deal with that. So one really active area of research is all these fungal microbial processes we've been talking about all night. Can we use that? Can we capitalize on that in some way to help trees cope with climate change or restore forests? And so again, I wanted to highlight Cassandra again because research stuff she's done with me. There's this, again, more cute puns. There's a saying we have, like, when climates change, species get mad, which means if you're a species and you live in a certain part of the world and your climate is changing, what are you going to do about that? You've got kind of three options. The idea is you can migrate, so you can move in space to track your climate, right? I'm going to move up north, I'm going to move up the mountainside. You can acclimate or adapt. So acclimate is change physiologically to tolerate the new climate. Adapt is change genetically or evolutionarily. Or if you can't do any of those things, then you die, right? Or go extinct. And so with this framework, if we look at where species are in the world and how climates are changing, this is where we get these really scary headlines about millions of plant and animal species going extinct because of climate change. It comes from this mapping process of saying, this is where it lives now. This is where that climate's going to be in the future. Can they track it? Or is that climate just going away? It's not going to be anywhere. If it's not going to be anywhere, or if it's going to be too far away from them, then they're going to go extinct. But we've been thinking, like my lab group and others, that trees and plants, because of these connections they have with their microbial associates, they may have another option. They're not so good at evolving. Trees don't evolve real fast, kind of slow organisms, long generations. So they're not going to, and they also don't move real fast. So those are not good options for trees, migrate or adapt. So that's why we're thinking about this other, sorry, ahead of myself. Could they do something else? Could they, by changing who they associate with, which fungi they're hooking up with, can they gain additional tolerance to changing climates? That's the theory, that's the question. And I'm gonna skip some of this. We did a big experiment. Basically, we take seedlings, we inoculate them with microbes from different places, put them back out there in the world, stress them out with these rainout shelters, and see if we give them microbes from one place versus another place, can we get them to survive drought better? Can we get them to survive winter better? Based on nothing else has changed about the tree, it's genetically the same trees, but we've given them different microbes. And so this is what that kind of data looks like. This is survival of trees here. And this is weird. This is the climate that the microbe came from, not the tree. All these trees are growing in the same place, but if they got microbes from colder places, they survived winter better. That's what we found in that experiment. So we were calling that this microbially induced cold tolerance. Something about the microbes from cold places was transferring something to the trees to help them survive winter. Let's do that. Then in our southern side, we did that up in northern Wisconsin, pretty cold up there. We also did this in central Illinois where it's pretty hot. If you're in central Illinois, it worked the other way. The trees that survived down there were the ones that got microbes from the hottest places. And so that was like this microbially induced heat tolerance we're studying. And then we made things worse for the trees by putting up these plastic shelters over them to block the rain. So we created drought over these seedlings. And if you were one of our seedlings living under the drought shelter, this is where we saw our strongest effect, this red line here. You did much better if we gave you microbes from drier places. So if we inoculated you with microbes from a dry place, you survived our drought treatment. If we inoculated you with microbes from a wetter place, you were toast. I mean, again, we haven't changed anything about the tree. They all grew in the greenhouse for 2 months with these microbial inocula, and then we planted them out in the field. And so, yeah, this was like really in that second summer where things got kind of hot and dry. We saw this really divergence between the ones that had microbes from dry places, doing great, microbes from wet places, all dying. So we're following up with that in 2 different directions now. One of them is much more like fundamental science. We're trying to figure out what in the heck is happening, which microbes are in these different inocula, and what are they doing to the tree that's making it magically tolerate drought. We think it has something to do with them accessing water in the soil a different way and then changing physiologically how the tree functions. So I've got some collaborators that know more about how plants work than me trying to figure this out. And then the other thing we're doing is taking that in a more applied or translational direction, saying, can we use that knowledge in some way? So I'm not going to go around inoculating the forest, right? That's not a reasonable thing to do. But we plant forest all the time. We're planting trees everywhere all the time. And when you plant a tree, most of you, if you've planted trees before, you're buying a tree, right, from a nursery. So that tree has grown for a while in some condition, and then you're taking it and planting it in some new place. That's how you do horticulture, but that's also how you do reforestation projects. These seedlings are grown in these outdoor nurseries, and then they're shipped to wherever they're going to be planted to grow the new forest. And so they're growing here for a year or 2 in these nurseries to get big enough to plant. So all that time, they're getting colonized by fungi. And one of the first things we're interested in is whether the fungi that they get colonized here, are they going with them to wherever they get planted, and are they sticking around? And then if they are they having good or bad outcomes for that tree? And can we do anything about it? Can we get them colonized with the fungi that are going to be helpful instead of harmful? So I won't belabor that, but we did some research that documented, yes, they're definitely getting colonized by fungi, different ones in different nurseries. And those fungal colonizers persist for at least 3 years after we've planted them in the forest. We can still find the fungi from the nursery on the roots. So it's not an ephemeral thing. But one question was whether that mattered for the seedling success. And so this is that kind of data we'd look at here. This is each of these dots is a bunch of different seedlings coming from different nurseries. And we're looking at what kind of fungi they have in their roots. It's on the x-axis here. In this case, it's how much of those ectomycorrhizal fungi, did they pick up from the nursery before we planted them? And then over here is how likely were they to survive in the planting? So if they started out life with a lot of these ectomycorrhizal fungi, they were surviving better, and especially if they had a higher diversity, so more different kinds of these fungi, more different species of fungi in their roots, they survived better. And if they had a lot of pathogens, that was bad. And then I'm gonna run out of time here, but we're also working with nurseries to switch what they're doing in the nursery to see if we can promote the most helpful fungi. So this is our research plot where we're trying to grow a forest in this old cornfield. This used to be a cornfield as of like a year ago. Now it's gone to pasture, as you say, and now we're planting trees in there. So trust me, there are a bunch of trees out there. You just can't see them. And here's a really complicated graph. But this is looking at how those trees survived based on what we did to them in the nursery. And I'll just skip, that's not so important. This is important. So in the former cornfield, where you think that the microbes in that cornfield have been beat up by decades of high input agriculture. If you're trying to put a tree in there, it works much better if that tree was not dosed with fungicide in the nursery, which is something that happens generally. The soils are fumigated, with chemicals to sort of knock back fungal pathogens, and that makes them grow faster in the nursery, but it actually made them survive terribly when we planted them in the field. Then we did our own fungicide treatment right before we planted them to try to nail that down. And the important thing was that these red arrows, if we added fungicide to the ones that did not get fungicide in the greenhouse, if we then added fungicide, that made things worse. And so the quick take home, I've talked too long already, is that, so we think we're working this up, developing healthy, diverse fungal associations in the nursery can help these trees establish, especially in soils that have been degraded by agriculture. And that's a huge amount of land in the United States that's getting abandoned from agriculture every year and then being converted back into forest reforestation efforts. So it's a big target. If we can make that work or make it work better, that's enough acreage to make an impact on the world. All right, and last thing before I release, I did want to talk about my little call to action here, our citizen science work. So the other thing we're trying to do is map what fungi live where in the temperate forest. So we know where the trees are. We know where trees live because the Forest Service has been mapping that forever. And they're easy. They're there. You can look at them. Knowing where fungi live, tougher. You can look for mushrooms. I was talking to someone earlier about that, and there's a lot of interest in surveys of mushrooms, which are great. But the vast majority of fungi don't make mushrooms. They don't make anything above ground that looks like anything. They live their entire lives underground, pretty much invisible. So you can't tell where they are by looking, but we can tell where they are, who they are in our laboratory. So we use DNA markers to identify fungi based on their DNA. And so we're running this project. We're looking for anyone and everyone, who's willing to dig up some tree roots from a forest and mail them to us in Wisconsin. And we have all the protocols, and we'll send you shipping labels and everything. We try to make it easy. We've had 5-year-olds do it. Probably not the mailing part, but then they come to us in Wisconsin, and then I teach a course in the spring where I have the undergrads in my course as like the lab project for their course. They work through all these roots, and they use the molecular tools to identify what fungal species are on the roots. Then we put it all together into these big data sets and try to answer questions. So here's where we've gotten, to get some sense of the scale of where we've gotten samples so far. I've been running this for a long time. We've had over 900 samples sent to us. It's way longer than 6 years. That's outdated, sorry. 5-year-olds to 95-year-olds. Broad range of people have been involved. You can do one root, takes 5 minutes. We've had some master naturalists in Illinois who are like our superstars that send us like 100 trees a year. And we use that data. I'll just show a little bit of trying to figure out, we look at trees across their climate range. Who are they hooking up with fungal-wise? Are they using different fungi at different parts in different climates to help them tolerate those climates? So we got there's some interesting patterns here with these non-mycorrhizal endophytes, which are our most mysterious fungi that are really connected to living in dry places. So trees living in dry places are investing a lot in these fungal interactions that we don't know much about. So it's opening up a lot of research questions for us. Those mycorrhizal connections I talk about, those are going in the opposite way. They use those in wetter, colder places. You're not supposed to get much out of this graph other than to say that we can also look at individual fungi, right? So this is a display of like each one of these dots is an individual kind of fungus. And it's showing how it associates with climate. Is it one that trees use a lot in hot places or one that trees use a lot in cold places? We could look at dry or wet. And the point of all of that is where we're trying to go with this, right? If we can build up a big enough data set from these volunteer samples, we can figure out which trees are using which fungi in which conditions. Then we do our experiments to tell us which of those fungi are leading to better outcomes for those trees. Are they providing that tolerance? And once we know what to aim for, then we use our collaborations with nursery growers and with restorationists to try to use that to grow a better forest. All right, my final message for the group. Next time you're out in the woods, consider looking down. There's a lot going on down there. I hope that was interesting. And thanks for people and taking questions.
Thank you so much, Rick. That was really fascinating. If folks want to come for the microphone.
Thank you.
Last time we had to have the speaker repeat questions. We think we've got that figured out so that the Zoom audience should be able to hear people asking questions with the microphone. So let's try that.
Yes, as far as collecting data, like you were just telling about, do you have any way of mapping out where the blank spaces are, where you're not getting data, where you could say on your website or whatever, we need
Yes.
stuff from here, and you have plenty from there.
I mean, the short answer to that
I'm
is,
sorry.
oh,
You're gonna
repeat the
need
question.
to repeat the question.
so the question, if I could paraphrase it, was asking whether there's areas that for our sampling, our citizen sampling, where we need more samples, or where you can go on our website and see areas that are undersampled and where we have a lot. The short answer is that we never have enough, even from the places where we have a lot, because we're also trying to track change over time. So we got a lot of samples from Illinois, but that's good because we have now a 10-year time series of them. So we'll never say no to a sample. But we got the central Midwest pretty covered because I'm based in Wisconsin and Cassandra's based in Illinois. So we sampled those two states well. We could definitely use more from over here. So New York for sure, anywhere around here. If you've got cousins in Indiana or Ohio, it's a bit of a dead zone for us. Southeast, we have to talk. Shocker to anyone. Florida's quarantined for soil shipment. You're not supposed to send soil or things with or even roots with a bit of soil them from the Southeast because they have too many exotic bad things down there. So we can do it, but it takes some coordination. Whereas anything up here, no rule, there are no rules. You can send things to us. Not from Canada. Crossing the border is also something we would have to negotiate with permits. Yep.
Yes, just read that a scientist studying this how fungi can benefit forests had her funding cut.
I'm not shocked. I don't know. We're still doing okay with funding in my lab. Yeah, part of it is, well, I'll talk away from the mic. Maybe when the Zoom call is not recording. I have thoughts.
Do gain of function mutations occur in fungi so that they can jump to new hosts?
I mean, presumably over evolutionary time, yes, that must have happened. So sorry, to repeat the question. He asked if gain of function mutations occur in fungi that allow them to jump hosts. So whether you're a pathogen or a mutualist, you do have to have there's sort of this complex chemical song and dance that goes on between the plant and the fungus in order to allow that interaction to happen. And so that's why you get a lot of specialism where like this fungus can infect this plant and no others because it's figured that out for that plant. So when there are host jumps, there has to be a mutation somewhere. It works a little differently for the pathogens than for the mutualists. One of the funny things, this is tangential to your question. I said most pathogens are specialists. So for the most part, a single fungal species infects one or a few related plant species. There are exceptions, but that's the dominant pattern. Whereas the mutualist, especially those arbuscular mycorrhizal fungi that have been around for 400 million years, there are only about 1,000 described species of them, and they interact with over 200,000 described plant species. So those are very generalized, meaning almost any mycorrhizal fungus will associate with almost any plant. And that's a result of like the sort of alignment or the conflict of interest. So with the pathogens and plants, it's an arms race. The plant's always evolving mutations to recognize pathogens and kick them out. That's how their immune system works. So the fungus has to evolve these gain of function mutations to overcome that recognition by the plant. So what works for one plant doesn't work for the next plant. And so they become specialized. For the mutualists, the plant, quote, wants them there. Its fitness is benefited by their presence. So they're under no selective pressure to recognize and kick them out. So those fungi have not been under selective pressure to evolve these countermeasures. And so they're left with this very generalized interaction where almost every plant wants them. Sorry, yeah,
Is there a danger of someone producing data?
Talk about funding cuts. In the last few months, I've gotten a lot of emails from the Department of Defense trying to build up their bioterrorism protection thing because they're worried about agricultural bioterrorism, right? As I said, there aren't a ton of fungal pathogens of humans. If there are medical doctors in the room that would know more about that than me. They're not a great target for like for a human disease, but they'd be a great target if you wanted to wipe out the agricultural system of a country.
Yeah, so if I understand correctly, each tree has only one kind of beneficial fungus. Is it ever happened that both of them?
Yeah, and I may have been too right. I may have been a little glib there. Each of those categories, the arbuscular mycorrhizal fungus and the ectomycorrhizal fungus, is a category with many fungi in it. So there are, I said, thousands some of the arbuscular mycorrhizal fungi, and so a tree can hook up with many different species in that category. It could have 5 or 10 different AM fungi. The ectomycorrhizal category has something like 7,000 described members, and there are probably a million more that are identified. And again, trees hook up with many of them at once. In fact, there's good evidence of having a diversity of associates, even a single tree having multiple different species of fungi in its roots is helpful because they can complement each other. There are a handful of trees, cottonwoods, aspens, willow trees, that will simultaneously make the arbuscular connection and the ectomycorrhizal connection. It's very rare, but it does happen with that group of trees. And then like eucalyptus trees in Australia are known to do that as well. That's an interesting phenomenon. We're not sure if that is a stable, like evolutionarily stable strategy where they're getting the benefits of both, or if it's a transition state. So all of the ectomycorrhizal relationships, their ancestors did the arbuscular mycorrhizal thing, and they evolved away from that towards this new phenomenon. It's happened independently multiple times in plants, usually long ago. So we don't watch it happening in real time, but these dually colonized species, some people think they're on their way, moving from sort of the old system to the new system. And maybe in a million years, they'll just be doing the ectomycorrhizal.
Since one process generates the nitrogen and the other one does phosphorus.
So yeah, so what, if I repeat that question? Yes, I don't know if I repeated it originally. So we're talking about, are there any trees that can form both kinds of mycorrhizal connections that I talked about? And yes, there are a few. They're pretty rare, and they do have different benefits. So one idea is that the trees that can do both actually are getting both of those benefits, the nitrogen and the phosphorus. Why doesn't that happen all the time? Is another question then. Like, why did everyone do that? One is that there's no free lunch in the world, right? So the plants are paying for this service that the fungi is providing. So they have to feed that fungus their carbon, which they could be using to grow. So they're using it to feed the fungus. And so they only do that if what they're getting in return is valuable. Nitrogen and phosphorus are both valuable for plants, but generally it is generally the case that in most situations, only one or the other is limiting to the plant. So they'll be in a situation where there's not enough nitrogen to grow. And so adding more phosphorus to that soil doesn't help them because they need them all, right? In a certain defined ratio. And then there are other parts of the world where there's not enough phosphorus to grow. And so adding more nitrogen doesn't help the plant. And so the situations where they would need both of those, more of both of those things are actually kind of rare. Maybe one reason why evolution has tended to go either one way or the other with these relationships.
Is anybody trying to raise funguses with specific properties commercially or or, or academic?
Yes. So the question is anyone trying to raise fungi or cultivate fungi or breed fungi for certain traits that would be beneficial? So I'll break that down. Many people are trying to cultivate fungi. I mean, people are cultivating culinary mushrooms, obviously, but there are also companies trying to cultivate inoculant products, we call it. So like you can go, actually they exist. You can go to Home Depot or whatever, like home and garden store, or you can go on Amazon. You can buy yourself a bag of mycorrhizal fungi, spores that you can dump in your garden or whatever. I don't often recommend it, partly because they're out there already for the most part. Like these fungi are everywhere. Find them in Antarctica. So to add more to your garden, the only reason you would do that is if you had some reason to believe that there weren't enough there already, which can happen, right? If you are maybe working with a really sort of depauperate topsoil, or you're starting from scratch with like potting mix, there's probably not any of these fungi in your like bag of potting mix. Or in the case that I talked about with like the restoring the former cornfield, like there are some fungi in there, but maybe not all the ones we want. Maybe it's a lower diversity of them. So that's where an inoculation product could be helpful. Problem is, it's like the nutritional supplement world in that it's not regulated in any way. So you have no guarantee that what you're buying is what is sort of on the bag. A lot of my colleagues who work in this, we've done a number of studies where we're like, well, it'd be really convenient for me, just for my own experiment, if I could buy some of this stuff in a bag. And it never seems to work. Most of the time it's dead. Like almost all the products we've tried to work with, they're quite delicate. And so it's a little bit challenging to like store them in a viable state, unless you sort of really know what you're doing. And some of the companies do, but some of them don't. So my general recommendation is not to do it. But I'm trying. I know some farmers who really swear by it, but I'm kind of a little skeptical. And then trying to make them better, that's a little clear. Most of what's happening, I think, is somewhat intentional, somewhat unintentional breeding for better inoculant products, like ones that aren't as delicate, ones that can survive in the bag longer, so that they can actually be a useful product that you get to in the back there.
It was one slide that you showed that it was the eastern half of the United States, and you were showing the growth of our muscular and the ecto.
The change, is it like the red and blue one?
Yeah.
Yeah. Let me see if I can pull it up there for you.
What's driving all of this, like down here in the Georgia, Alabama?
I probably, this paper, which again, I should say, not my research, this is just a graph I pulled. It's just looking at net changes with these forest surveys. So all sorts of things can be happening, but most of this is probably pine plantations. So pine trees would be blue here, ectomycorrhizal. And for the paper industry, there's a lot of loblolly pine plantation in this part of the country. And so that's been growing, and that shows up as an increase. It's not necessarily that they're taking over a native forest, other than like it was cut down and then this plantation was put there. That's a good question. I don't. Oh, sorry. The question was, how did the chestnut blight get introduced? I don't know for sure. And I also don't know if it is known for sure. I'm trying to debate whether it's just I don't know it or nobody knows it. Probably on wood, the way a lot of these forest pathogens move around is like timber shipments, because they're living inside of the wood tissue. And then, so I don't know if you've ever been camping and you've seen signs up. Maybe they don't have them out here. They say, "Don't move firewood. Don't move firewood, please," because that's where a lot of things can move. Both fungal pathogens and insect pests, like the emerald ash borer, move around in timber and firewood. So especially untreated lumber, like the raw logs, that's where that's going to be full of stuff. Yeah.
Is there any evidence that trees have ever try to grow, to be able to feed themselves without these fungi?
Yes, that's a great question. So the question is there any evidence that trees have tried, I mean evolutionarily, to like pick the habit, right? And live on their own? Yes, well, plants broadly. There are species of plants that don't do this, genetically don't do this. They've lost whatever gene is necessary to make these connections. And they do okay. The biggest group of plants would be the mustard family. So cole crops, broccoli, mustards, all of the Brussels sprouts and all that stuff. Do not make this stuff. Do not make these connections. A little chicken and egg. You know the spicy taste of wasabi, horseradish, comes from a chemical called the glucosinolate, which is a unique chemical that these plants make. It is a deterrent, if you imagine, to herbivores, to things eating the plant. As humans, we eat this stuff. We like it, but most animals don't. It's also antifungal. The chemical is also antifungal, so it's a protection for the plant against fungal pathogens. But it's, I say it's a chicken and the egg because we're not sure which was first. Those plants can't make these mycorrhizal connections because they make that chemical. But the question we don't know is, did they start making the chemical and then lose the reaction? Or did they lose the interaction and that allowed them to evolve this chemical, right? Because they didn't have anything to lose anymore. It happens rarely. What does happen, and this is true in crops, where this has been well documented with breeding crops, plants, in some ways, there's some evidence that plants have been on a 400 million year long journey away from these arbuscular mycorrhizal connections. So they crawled out of the ocean as algal cells trying to live on land, which is hard to do, and these fungi helped them do that. They had no roots, right? Mosses don't have roots. As you look through the evolution of plants, to mosses, to horsetails, to ferns, to pine trees, to oak trees or whatever, root systems are one thing that has changed a lot along that evolution. As plants have lived on land longer and longer, they've developed better and better root systems. Finer, skinnier roots that are better at absorbing things from soil. And so there's one sort of rule of thumb that plants, although most plants do this, they don't all depend on it to the same degree. Some plants are extremely dependent on these fungi. They cannot grow without them. Others can take it or leave it. Like they'll do it, but if they don't have it, they'll still grow okay. So it's facultative, and we think that has a lot to do with their root system. As roots have gotten skinnier and skinnier, those plants don't need these fungi to the same degree. The ectomycorrhizal relationship I talked about works a little differently because there the fungus does something that no plant can do with these enzymes that can break down organic material and mine nitrogen. That no plant's going to evolve that trait anytime soon. It's more like, they might as well evolve wings. So that one's probably here to stay. And 100 million years, it'll still be here.
I'm going to take a few questions from the folks on Zoom now. So I am going to try reading them into the microphone and hopefully everyone will hear. Okay, so first question, is there a general fungal difference between native and non-native plants?
I don't need to read the question again, right? Because it's something, the most general statement I can make is that when we as humans move organisms across the world, more often than not, we don't move all of their associated organisms with them. So there's a hypothesis we call enemies release, that when we move invasive plants to new areas, they often don't have any of those specialist pathogens I talked about. Because they had them back in Europe or Asia, wherever they came from, but we didn't move them with them. And then the pathogens in North America are specialized on North American plants. So they can't infect this European plant or this Asian plant. And we think that's one key reason why exotic plant species can be so successful and kind of take over. I used to study garlic mustard a lot. I don't know if you guys do that around here. That's one where it's got a lot more things attacking it and eating it in Europe than it does here. Same with Japanese barberry or all of those things. So that's sort of an answer to the question. Generally, they have less fungi, lower diversity of fungi sort of attacking them. What is kind of funny is that those helpful fungi, the mutualistic ones, I said, those are often very generalized. So the mycorrhizal fungi in North American soil will connect with that European plant or that Asian plant, but the pathogens in the North American soil probably won't infect that European plant. So they get all the good stuff and none of the bad stuff. That's sort of how one way they're able to take over.
Great presentation. You touched briefly on the possibility of mother trees nursing offspring, but you also mentioned that fungi in the soil prevent dominance of any particular species. Aren't these two events at odds with each other?
Yes, they are. Yes, they are. And that doesn't mean they can't both be true, because it's a complex world out there. And so it can come down to the balance, whether there's like mother tree. I'm a little skeptical of the like mom to offspring thing, but definitely like the adult tree to seedling tree of the same species. The common mycorrhizal connection is definitely a possibility there, which could be a net benefit. But then I also talked about all those pathogens that adult mother tree has accumulated in her roots. That would be a detriment, right? One thing where we do see, and I think this is, again, a little bit speculative, if you're in the tropics, where there's that huge diversity of tree species, right? Those are all making that arbuscular mycorrhizal connection. Those arbuscular fungi don't make very big networks. They just don't grow very far in the soil. So the mother to offspring thing is much more with the ectomycorrhizal fungi out in the conifer forest. So the listener picked up on a really actually key idea, that diversity in the tropics, we think that's really pathogen driven. There isn't much of this common mycorrhizal network stuff going on there because they're all doing the AM strategy. And so that pathogen process dominates and you get the little zones of death and you get the high diversity. If you go to the conifer forest, especially those Western ones that where the stuff has been studied, those Douglas fir things, it's all Douglas fir. They're very low diversity forests, one dominant tree. And so it's the opposite, right? There's dominance of this sort of mycorrhizal connection that would tend to make it hard for any other tree species to get into that, right? If you're not part of the network, you get kicked out. Why a pathogen hasn't wiped out that forest, I don't know. They must have other strategies. There is some evidence that the ectomycorrhizal phenomenon in general is good at protecting trees from pathogens because of the way they grow.
Is the new knowledge about fungi allowing for a more intelligent use of agricultural fertilizers and herbicides?
Could it be? Is it going to be? I could talk for a long time about the other half of my research. I talked all about my research that we do in forests. We also work in agricultural fields. I work with a lot of potato growers and some other things. Certainly there's always, it seems to come up again, even in my career hasn't been that long, sort of cycles through, the resurgence of, are we serious? Are we going to take sustainable agriculture seriously, or regenerative agriculture, organic? Every new name lasts a couple of years before it gets kind of dropped and a new word comes up to take its place. It's a pretty small percentage of the acreage, if you will, on the world, but a lot of people are doing it and they're successful at doing it, growing in a more lighter impact on the planet. I get a lot of interest from organic growers about soil health, soil biology, these mycorrhizal inoculant products, which in some ways I feel like I'm a professional wet blanket when I go to those meetings. Everyone's really excited about them. I'm like, don't buy them. But I'm a big proponent of, what I tell my farmers that I work with is, you have the microbes in your soil that you have for a reason. It's because if you don't have the ones you want, it's because the conditions are not right for those microbes to grow and live there. And so like buying them in a bag and putting them on your soil isn't going to help anything because they're just going to die because there's too much tillage, there's too much chemical inputs, too much fertilizer or whatever. So you have to change. It's tough because you kind of have to change the system first to make the soil hospitable to these microbial populations. And then maybe you get the benefits from them eventually. But like try telling a farmer, like, you got to lose money for 5 or 10 years before the stuff picks up. Like the farm's not going to be there anymore. It'll be out of business before that happens, right? So one thing that we as researchers are trying to do is figure out, are there ways to like jumpstart that? Okay, what can we do to try to get over the hump faster? The biggest, I don't know if you guys are very familiar with organic agriculture, but one of the biggest challenges for like starting a new organic farm or converting from a conventional farm to an organic one is that you have to go 3 years farming organically before you can sell your products as labeled organic, right? And that's to like get rid of the residues of pesticides or whatever. But that means you're losing money for 3 years and not getting the price benefit. And that alone is enough to make it really hard for farmers to do. So that's one of the things that prevents more organic acreage from being there. It's like, oh, they could be profitable if you got them, you know, to the promised land at the end of that 3-year period. But farming such a low margin business that it's hard to go 3 years and lose money.
The Natural Areas Conservancy in NYC convenes a 21-city national network called Forests in Cities. I think our partners would be interested in providing samples for the forest fungi product.
That'd be great. All right.
And the last one from online, would you have the same hesitation accepting urban samples as Floridian samples?
No, the Floridian samples, that's a legal thing. What we've put up on our website, if you go to it, in terms of like where to sample, we were struggling as a research team, Cassandra and I and Katie, the grad student, of what sort of line do we want to draw? Will we take a sample from any tree, or do we want to draw some line? Do we want trees from people's backyards? And right now, our line is that if the grass is mowed around the tree, don't sample it. But if the grass isn't mowed, that's natural enough for us. We're not trying to be like, really strict about what is natural, right? It's not a word that has a lot of real meaning. I do think urban trees, in terms of like a city street tree, right, are in a little bit different situation just because of the soil that they're growing in and the history of that soil and the history of the tree. It's not, we do want to study the microbes that trees would accumulate sort of themselves as they develop in a place. So if it's a tree that's sort of been planted as an adult tree and then you sample it the next year, it did not recruit its own microbes in its development, right? But a tree in Central Park, if you had permission to sample there, would be great, right? That's in the rumble gramble part, right, where the trees are sort of doing their own thing. So yeah, we're down for urban trees if they're not an isolated like street tree, but if they're in some part of the grove. I do think maybe we'll expand it someday. Somebody should. There has been a little bit of research on urban trees and their fungal connections. I'm not an expert on it, but there should be more work on it.
Are these fungi transferring any breakdown products from microplastics into the tree?
So the question for Zoomers, are fungi transferring microplastics into the tree? I have never thought of that, and I don't know. Microplastics are very small, but when we talk about things that these fungi are transferring, it's like dissolved. It's more like, I think it's, yeah. So possibly, depending on how broken down it is, have you broken down to like a dissolved chemical, but which they could be like a PFAS or something, right? The forever chemicals. I don't know. I don't know if people have measured that in plant tissue, probably. I mean, they're ever else. I would be shocked if not in plant tissue. If they're dissolved in water. Plants are not completely passive entities in the world. They do have mechanisms in their roots. They don't just suck the water straight up. It goes through cellular processes where they can differentiate things. So it's possible that it can't get past what we call the Caspian strip, which is a part of the root anatomy that all water has to pass through this, don't they just flow? It has to pass through these cells, and the cells keep most of the bad stuff out. But these are novel chemicals, so there's no guarantee that the plant sort of evolved defense mechanism against external chemicals would work against them, right? I should look that up. Somebody might know, I don't know.
Right. We're going to have to stop. Thank you, Rick. This has been absolutely fascinating.